Assalamu alaikum. I am Dr. Sharmin Rahman, Associate Professor, Department of Anatomy at the Newszonebd Medical College. Today my topic is muscle tissue. Muscle tissue is composed of cells that are specialized to shorten in length by contraction. Muscle cell is called myocyte, and myocytes are elongated in one direction and are therefore referred to as muscle fiber. Microfilaments and associate proteins together generate the forces necessary for cellular contraction, which drives movement within certain organs and the body as a whole. Before going to the histological structure of muscle tissue, we have to know about some terminology such as sarcolemma, which is the muscle cell membrane, and sarcoplasm, that is the cytoplasm of the muscle tissue. Sarcoplasmic reticulum, these are the smooth endoplasmic reticulum of muscle tissue. The sarcosomes, these are the mitochondria of the muscle tissue. On the basis of morphologic and functional characteristics, the muscles can be divided into three types: skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is composed of bundles of very long cylindrical and multinucleated cells that show cross striations. Their contraction is quick, forceful, and usually under voluntary control. Cardiac muscle also has cross striations, and these are composed of elongated branched individual cells that lie parallel to each other. At the sites of end-to-end contact, that there are presence of intercalated disc that is only found in the cardiac muscle. Contraction of cardiac muscle is involuntary, vigorous, and rhythmic. Now smooth muscle, these consist of collection of fusiform cells that do not show striations. Their contraction process is slow and not subjected to the voluntary control. There are subcoverings in the skeletal muscles, that is epimysium, perimysium, and endomysium. The entire muscle is surrounded by the epimysium, that is a dense irregular collagenous connective tissue. Perimysium, a less dense collagenous connective tissue derived from epimysium, surrounds bundles or fascicles of the muscle fiber. Endomysium, composed of reticular fibers and an external lamina or basal lamina, surrounds each muscle cell. In this figure, we can see this is one muscle fiber that is surrounded by the endomysium, and some muscle fibers that are surrounded by the endomysium forms a fascicle that is surrounded by perimysium, and some fascicles that form the whole muscle that is surrounded by the epimysium. There is another picture where we can see this is muscle fiber surrounded by the endomysium, this is perimysium, and the entire muscle is surrounded by the epimysium. Now microscopic structure of the skeletal muscle. As observed with the light microscope, longitudinally sectioned skeletal muscle fibers show cross striations of alternating light and dark bands. The darker band are called A band, that is anisotropic or birefringent in polarized light. The lighter bands are called the I bands, that is isotropic, that do not alter the polarized light. In this figure, we can see this is A band and this lighter zone is I band. In the transmembrane electron microscopy, each I band is seen to be bisected by a dark transverse line that is known as the Z line. The region of the myofibril between the two successive Z line is known as the sarcomere. It is considered to the contractile unit of skeletal muscle fiber. The eye bands consist of the portion of the thin filaments that do not overlap the thick filaments, and the A band is composed mainly of thick filaments and in addition by the overlapping thin filaments. A band shows the presence of lighter zone in its center that is known as the H zone, and this H zone is bisecting by a line that is known as M line. This is another figure where we can see this is a myofibril, where we can see this is A band and this is I band, and in the A band there are composed of thick filaments and thin filaments, and in the I band only thin filaments are present, and in the I band there is a bisecting line that is Z line or Z disc, and in the A band this is lighter zone that is known as H zone, and there is a line known as M line. These are thick filaments and these are thin filaments, and this is Z disc, and between two successive Z discs, the area between two successive Z discs is known as sarcomere, which is the contractile unit of skeletal muscle. To provide for a uniform contraction, skeletal muscle fibers have a system of transverse tubule or T-tubule. These finger-like invaginations of sarcolemma form a complex network of tubules that encircles every A-I band boundaries of each sarcomere. Adjacent to opposite sides of each T-tubule are expanded terminal cisterna of the sarcoplasmic reticulum. These specialized complex consisting of a T-tubule and usually two small cisternae of sarcoplasmic reticulum is known as triad. In this figure we can see this is T-tubule or transverse tubule, and on each side of the T-tubule there are presence of terminal cisternae of sarcoplasmic reticulum, and this unit known as triad which is present in the AI boundaries or AI junction. Now cardiac muscle. We already know that cardiac muscles are branching fiber and they exhibit a cross striation, and each cardiac muscle cell possesses only one or two centrally located pale staining nuclei and presence of dark staining transverse lines that cross the chains of the cardiac cells at irregular intervals known as intercalated disc. In this figure we can see this is a cardiac muscle fiber which shows the branching, and these are nucleus which are centrally placed, and this is intercalated disc. Now intercalated disc, it has a transverse region and longitudinal region. The transverse region of these step-like disc have many desmosome and fascia adherents. These fascia adherents in the epithelial tissue known as zonular adherents, and together these two junctions serve to bind cardiac muscle cells firmly together to prevent their pulling apart under constant contractile activity. The more longitudinal portions of each disc have multiple gap junctions, which provide ionic continuity between adjacent cells. These act as electrical synapses and allow cells of cardiac muscle to act as in a multinucleated syncytium, with contraction signals passing in a wave from cell to cell. In this figure we can see these are gap junctions and these are desmosomes. So these desmosomes are present in the transverse region and these gap junctions present in the longitudinal region of the intercalated disc. This is another figure where you can see these are gap junctions and these are desmosome and fascia adherents. The sarcoplasmic reticulum of cardiac muscle does not form terminal cisternae. Instead, small terminals of sarcoplasmic reticulum approximate the T-tubules these partners resulting a diet. Now, smooth muscle. These are elongated, tapering and non-striated cells, each enclosed by a thin basal lamina and a fine network of reticular fibers. Each cell has a single nucleus located in the center of the cell, and a rudimentary sarcoplasmic reticulum is present, but there is no T-tubules. Now, what are the comparison between the skeletal muscle, cardiac muscle and smooth muscle? Sarcoplasm and myofibrils that are present in the skeletal muscle and cardiac muscle but not in the smooth muscle. Nuclei, skeletal muscle are multinucleated and peripherally placed. Cardiac muscles are one or two nucleus present and centrally placed, and in the smooth muscle there are single nucleus which are centrally placed. Sarcoplasmic reticulum are well developed with terminal cisternae in the skeletal muscle and poorly defined with some small terminals in the cardiac muscle, and in the smooth muscle there are some smooth endoplasmic reticulum. T-tubules present in skeletal muscle, small and involved in triad formation, and in the cardiac muscle they are present large and form the diad formation, and there are absent of T-tubules in the smooth muscle. Cellular junction present in the intercalated disc of the cardiac muscle, and there are nexus or gap junction present in the smooth muscle. Contraction. There are voluntary contraction in the skeletal muscle, and the smooth muscle and cardiac muscles are involuntary in nature. Calcium control. Calcicoestrine in terminal cisternae present in the skeletal muscle, and in the cardiac muscle calcium from extracellular sources and the sarcoplasmic reticulum, and in the smooth muscle calcium from extracellular sources and the sarcoplasmic or endoplasmic reticulum. Calcium binding proteins, troponin C present in both skeletal muscle and cardiac muscle, and there are calmodulin present in the smooth muscle. Regeneration present in skeletal muscle via satellite cells and absent in cardiac muscle and present in the smooth muscle. Mitosis only present in the smooth muscle. Nerve fibers, there are somatic motor supply in the skeletal muscle, and there are autonomic supply in the cardiac and smooth muscle. Connective tissue, there are presence of epimysium, perimysium and endomysium in the skeletal muscle, and connective tissue sheaths and endomysium in the cardiac muscle, and connective tissue sheaths and endomysium also present in the smooth muscle. These skeletal muscles are long, cylindrical and many peripheral nuclei present, and cardiac muscles are branched cells, intercalated discs are present, and one or two nucleus are present. And in the smooth muscle there are fusiform cells with no striation and single nucleus that is centrally placed. So that's all for today. Thank you.